In this video, we're going to talk about the structure and the function of proteins. And once again, most of the diagrams and examples that we will explore are found in our Cognity higher level textbook. So the question is, what are proteins? They're actually what DNA codes for. So DNA is the genetic material, and it contains all of the instructions for building all of the proteins within our body. 50% of the mass of all cells are actually protein. And proteins play a structural role, so they provide support and structure, and they also can play a functional role, so they actually can do physical jobs within a cell. They are amino acid polymers, so they're made of many amino acids joined together. There are 20 amino acids, and 8 of them are essential to our diet, meaning that our bodies can't build them from their component parts. We have to actually ingest them through our diet. First of all, enzymes are biological catalysts. So we're going to spend a whole lesson just on enzymes in the lesson following this one. Immunoglobulins are proteins that fight disease. So when we're in our homeostasis unit and we learn about the immune system, we'll learn about immunoglobulins. You've probably heard of hemoglobin before. It's a protein that's found in red blood cells that transports oxygen. Keratin is important in the structure and function of fingernails, hair, and hooves, and so on. Fibrin is a protein that helps our blood to clot so that we can form a scab and prevent ourselves from bleeding too much. And then collagen is part of your bones and skin. These are just a few examples of the many different proteins within our bodies. So one skill you need to be able to do is draw a general amino acid. And there is a central carbon. It's also called the alpha carbon. It's bonded to 4 different things. An amino group, and again, when you take organic chemistry in your chemistry unit, you'll learn that this is an amino group, or NH2. There's a carboxylic acid, or carboxyl group on the other side. And then there's a hydrogen. Every amino acid has an amino, a hydrogen, a carboxyl group, and then a side chain. We often call that the R group. And that's the thing that varies from the 20 different amino acids. The simplest amino acid, the R group or the side chain, is just hydrogen, and that makes glycine. We'll learn about all the other amino acids later when we do our molecular genetics unit. Amino acids are joined together through condensation reactions, just the same way as joining monosaccharides and lipids together. And there are 20 different amino acids, as I mentioned before. You don't need to memorize them. You may not even learn most of them or explore most of them, but they are present and you may come across them. So some, as I mentioned, some amino acids are essential. Some can be formed in the body, so they're non-essential. As long as you have the component parts, our body can build them back up again. But some amino acids can only be obtained by food. They're called the essential amino acids. And we need to ensure that we consume all the essential amino acids in our diets to ensure that we build the many different proteins we need for our body's functions. While meat is often considered the best source of protein, vegans can eat a balanced protein diet through lentils, quinoa, and so on. They just have to ensure that they are getting sufficient protein in their diet. That being said, even meat eaters could be missing some of the essential amino acids or some of the other important vitamins and minerals that are found in fruits and vegetables if they only ate meat. There are infinite number of combinations of amino acids and an infinite number of lengths of amino acids. So there are so many different potential proteins out there. So remember, I mentioned already that DNA contains the code for building all our proteins. First, a gene has to be transcribed into what is called mRNA, messenger RNA, and then it gets translated into a protein or polypeptide. The genetic code is made of codons, which are a set of 3 nucleotides. So remember, we have adenine, guanine, thymine, and cytosine. There are 64 codons for the 20 amino acids, and actually the codons are made of RNA. So it would be adenine, uracil, cytosine, and guanine. And because there are 4 different nucleotides, and they can be added in any combination, there's 4 times 4 times 4 possible arrangements, and that's what leads to the 64 codons. Polypeptides can be any length, and they can have any combination of amino acids. So like I said, there's an infinite number of possible combinations. So one thing you need to be aware of is some examples of polypeptides. So first of all, lysozymes. Those are antimicrobial enzymes that are found in your tears and saliva that actually act as a line of defense to prevent pathogens from entering our body through our eyes or through our mouth. We also have alpha neurotoxins. That's a component, we don't have it. It's a component in snake venom that affects our nervous system. We don't want to get bitten by a venomous snake. Glucagon is a pancreatic hormone that raises blood sugar when it gets low. We'll learn about it and insulin when we get a little bit later into our course. Myoglobin is a molecule that's in our muscles that binds oxygen and makes it available to muscle cells. It actually attracts oxygen better than hemoglobin. So when the oxygenated blood gets to the muscle cells, the myoglobin grabs onto the oxygen away from the hemoglobin. And then finally, titin is a massive protein that gives muscles their springiness, which is kind of funny that it's so huge but its job is to give muscles springiness. So let's talk about the properties of amino acids. As I mentioned before, there's 20 amino acids, and they can have different properties. They can be polar, they can be nonpolar, they can be acidic, they can be basic, and all depends on their R group. And then the peptide bonds link amino acids together. And remember, they're connected through condensation reactions. And then the 3D shape or the conformation of a protein depends on the amino acid sequence. So the polar regions will all clump together, and the nonpolar will clump together. The acidic regions may all interact with, acids and so on. So the folding of the protein has to do with the amino acids and the order that they're found in a protein. And a really small mutation that can change the property of one of the amino acids or substitute a different amino acid with a different property can actually significantly impact the protein and whether or not it can function as it should. Now, there are 4 levels of protein structure, and we're going to talk about that now. First, we'll talk about the structure of a dipeptide. So you need to be able to draw a dipeptide and show the condensation reaction. And again, you can just draw the amino acids with their R groups, not the individual ones. So what happens is 2 amino acids, so the N, the C, joins up with another, so the carboxyl group joins with an amino group of the adjacent one. So the way you write it would be NCC, NCC, with the carboxyl group sticking up here, the amino group down there. So the carboxyl loses the hydroxide or the OH, and then the amino loses one of the hydrogens. And this is what the bond should look like. That's the peptide bond. You would need to label that. You would need to say that this is a dipeptide, and you would need to draw this exactly as you see it to get full marks. So we're going to highlight the 4 levels of structure, and then we'll expand a little bit further. So primary structure is just the amino acid sequence. Secondary structure are when the amino and the carboxyl groups interact with one another, and you can end up with spirals called helices, alpha helices, and you can end up with these flat lined up regions called beta sheets or beta pleated sheets. The tertiary structure, this thing here, you're seeing that it's looking much more jumbled. That's a 3D structure of the polypeptide, and that's when the R groups all interact, so all the polar regions will stick together, all the nonpolars will stick together, and so on. And then finally, a quaternary structure is when you have multiple polypeptide subunits all forming together. An example of that would be hemoglobin. So again, the primary structure is the amino acid chain, and literally it's drawn like, a beaded chain. So each circle represents one amino acid. It'll start at one end with the carboxyl group, and then it'll end with the amino group. And each of the little links between the beads are peptide bonds. The circles are the amino acids. The secondary structure is when hydrogen bonds form between the carboxyl and amino regions of the polypeptide, and they can form alpha helices or beta pleated sheets, and we'll look at those now. So an alpha helix is when hydrogen bonds form between a carboxyl group on one amino acid and the amino group 4 amino acids away, and that keeps happening regularly, so you end up with this spiral formation or a helix. An example of a protein that has a lot of alpha helices would be keratin, which is in our hair. In beta pleated sheets, 2 parts of the polypeptide line up parallel to each other, and hydrogen bonds occur between the carboxyl groups and the amino groups of the opposite sections. And we represent that in a drawing with these flat arrows. And you can see that they're lining up anti-parallel to one another. So one arrow will point that way, the other one that way, and so on. So if you see these drawings, that's the beta pleated sheets. An example of a protein that has a lot of beta pleated sheets would be spider webs. Tertiary structure is when the polypeptide supercoils, and that's when the side chains interact with each other and their environment. And a few examples are listed. So there could be hydrogen bonding. There could be disulfide bridges, and that's when there's sulfurs in 2 different amino acids that form a covalent bond with one another. We'll talk a little bit about that later. Ionic bonding, so ionic bonding between a positive and negative ions. The hydrophobic groups will attract each other, and the hydrophilic groups will attract each other. Finally, quaternary structure is when 2 or more proteins fold together to form a functional protein, and the example here is hemoglobin. So there's actually 4 polypeptide chains that make hemoglobin up. Membrane proteins. So transmembrane proteins will have regions that are nonpolar because the middle part of the cell membrane, the phospholipid bilayer, is nonpolar, and then they'll have regions that are polar, so that will allow them to bend into the bilayer, and that'll help them be able to also interact with the extracellular fluid and the cytoplasm. One example of a membrane protein would be rhodopsin, which is a light-absorbing cell of the retina, which we can see in the diagram at right. So this is rhodopsin. Non-conjugated proteins are proteins that are composed only of polypeptide subunits. So collagen is an example. It's made of 3 subunits, and insulin is made of 2 subunits. And what this means is that these proteins don't have a glycoprotein, they don't have a carbohydrate or a metal ion or something else bonded to it. They're just made of polypeptides. Conjugated proteins, on the other hand, have other molecules or metal ions bonded to them, and that gives them specific functions. So again, hemoglobin, which we looked at a couple of slides before, has 4 polypeptide subunits, plus they have iron-containing heme groups, which let the protein carry oxygen. Without the iron heme group, the protein is unable to carry the oxygen. So denaturation is when a protein unfolds. So the hydrogen bonds or the ionic bonds or the disulfide bridges can be broken. And this can be altered by heat or pH or chemicals that are outside the ideal range for the protein. And when a protein denatures, it can't function anymore because it's not folded properly. And if the peptide bonds themselves are broken, or for some reason that protein can't refold, the protein has been irreparably damaged. And that damage is called coagulation. So I have a personal example of denaturation. So when I was growing up in the late 80s and early 90s, it was really popular to have permed hair. And I had a friend that had gone through beauty school, and she wanted to try out her newfound skills on me by giving me a perm. All I had to do was pay for the materials, and she would perm my hair. So what happens when you perm hair is you put chemicals on the hair to break the disulfide bridges that are found in your hair proteins. Your hair has been wrapped around these curlers, and you put the chemical on to break the disulfide bridges. You let that react, and then you put a second chemical to stop the reaction and reform the disulfide bridges. Perm solution smelled really bad because of the sulfur to reform the disulfide bridges. But what happened was my friend missed a step, and my hair did not curl as it was supposed to. So she did a very generous offer and said I wouldn't have to pay her again, and she re-permed my hair, and she doubly denatured it. And so for a whole year, as I waited for my hair to grow out, if I brushed it too hard, little pieces of my hair would break off because she had coagulated my hair. It was irreparably damaged. It could not have the texture and the structure that the protein should have had because she had applied chemicals that set my hair outside of its ideal range. It was a terrible year. So globular proteins. These are proteins that fold into an irregularly folded spherical shape. And these proteins are soluble in water, and they tend to act as enzymes, transport molecules, and so on. So one example of a globular protein is insulin, a hormone that regulates our blood sugar. Again, we'll look at the functions of insulin later when we learn about our pancreas and its role in blood sugar maintenance. Fibrous proteins, on the other hand, are more linear in shape, and they are insoluble in water, and they perform or provide a much more structural role in the body and within the cell, and they will form components of our skin, our hair, our bones, and so on. So one example is collagen. It's the body's most prevalent protein in the body. It's folded into a triple helix and provides flexible support to structures. Finally, you've probably heard of the genome. That's an organism's entire complement of genetic material. The proteome is an organism's entire set of proteins. So it's the unique set of proteins coded for by one's genome. And every species has its own unique set, but there's also variation between individuals within a species. Even identical twins. Even though identical twins have the same genetic makeup, their proteome can be different because the way their body expresses its genes may be slightly different from one person to the next.